Hello, and welcome to this video on water and water's interactions. So let's get started. Water has a chemical formula H2O, and we can sort of sketch this as an oxygen bonded to two hydrogens. These are single bonds. The oxygen also has two lone pairs of electrons. Now, oxygen is more electronegative than hydrogen. We could also say that it has a greater electron affinity than hydrogen does. So what this results in is there are more electrons flying around in the oxygen region of the water molecule than in the hydrogen region. And we want to make sure that we keep track of things like that when we think about molecules, especially water. So what we do is we say that it has a partial negative charge, and we write that using delta negative. The two hydrogens have a partial positive charge as a result because water is a neutral molecule. So we write delta positive near the two hydrogens. This is in contrast to a full charge like we might see on a chlorine ion in solution. That's a full charge here. These are only partial charges. So this results in a phenomenon that we call a dipole. Dipole is similar to, you could think of it like a magnet, and so there is more negative charge here and more positive charge here, almost like the north and south ends of a magnet. And opposites attract, so dipoles end up aligning or attracting each other. So when we have a liquid like water that is made of this, that is a dipole, it has these attractions called hydrogen bonds, also known as dipole-dipole attractions. And this is because the delta negative oxygen is attracting the delta positive hydrogen. And so it results in things like a higher boiling point of water and greater surface tension of water and other neat chemical phenomena like that. We call these attractions here hydrogen bonds in the case of water. So that means that there's additional work needed to pull water molecules apart. Like I said, water has a higher boiling point than than other liquids that are not dipoles like this. So now let's think about what happens when we have other things in water. So I'm going to pick the phospholipid sphingomyelin as our sort of test molecule. And so we want to know how this interacts with water. So it's important that we note the different regions of sphingomyelin. So let's write that in blue. So sphingomyelin has this non-polar region. And it also has a polar region. And it also has a charged region. So these different regions of this molecule, sphingomyelin, are going to sort of dictate how water interacts with it. It's important to note that just like in the case of water, where we had the delta negative and delta positive due to different electron attractions, these also exist within larger molecules or other molecules like this. So for example, this oxygen here is going to be delta negative. This nitrogen here is going to be delta negative. This hydrogen will be delta positive. And so when water gets near these, or this gets placed into water, this sort of breaks apart those hydrogen bonds that we were talking about, that I talked about earlier. These are our hydrogen bonds. So water wants to reform these because it's more stable that way. So what it will do is it will form these interactions not just with itself, but also with the molecule in question. So we'll have, for example, hydrogen here, bonded to an oxygen, bonded to a hydrogen, and there'll be this attraction between this oxygen and this hydrogen. There'll be an attraction between this hydrogen and this oxygen, and this is because the partial negative and partial positive charges in these regions attract. Now, in the case of this nonpolar region, there is no partial charge like this because carbon and hydrogen pretty much share electrons equally. And so as a result, the nonpolar regions like this sort of repel water. They don't attract them. And so these regions here are going to try and get away from water, as opposed to the polar regions, which are going to try and which will be attracted to water. Now the charged regions here also interact with water, so we're going to have plenty of hydrogen attractions to this, because hydrogen is the positive partial positive charge attracted to this negative charge here, and then here with the positively charged amine group, the oxygens are going to be more attracted there. And so what this results in is the hydrogen bond network of water reforming so that the this molecule will sort of be like restabilized. And again, there's lots of water, so these bonds will also form between the water molecules. So if we get enough of these, of a phospholipid like this in water, we're going to see regions that are sort of attracted, that sort of have water around them, and regions that don't have water on them. So if we sort of write this molecule, if we sort of generalize it as having this water attractive end with a circle and then a tail that's nonpolar like this, it'll result in a bilayer forming. And once I finish drawing, it'll make more sense. So the bilayer basically seeks to have water, lots of water here, and not very much water in the middle. So we'll have lots more water interacting with these charged or polar regions, and not very much water interacting with the nonpolar regions. And so this structure is what sort of defines cell membranes, and that's important when we're studying biology or biochemistry or things like that, because we just want to know how water might behave in the presence of other molecules. Now, I've used this specific sphingomyelin molecule to demonstrate how water interacts, but this is true of all molecules. Any place that there is a positive regions of positive charge, like this amine group, this hydrogen here, the oxygen will be attracted to that, and it'll sort of form a shell around that. And then any place there is negative charge, the hydrogens will be attracted to that. And so if we think about like a chlorine ion in solution, it's going to attract the hydrogens, just like this phosphate group did. And here is my works cited.